Is it doing it with the audio? Yes, 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 yes. Cool.
Recording.
Yes. Should be. Okay. All right. Thanks everyone for coming. If you're lost in this unit, it's okay. Because we're gonna go through like literally every single pathway today. Yago knows a lot. Yago's taking the MCAT, so he's a big guy. And honestly, like it's a lot of terms, but if you get the steps down of each pathway, like it becomes a little bit easier to kind of distinguish everything because you're not thinking it in terms of like this does this, but more of like this happens first and then this happens. So don't get too overwhelmed by all the terms. We're gonna go over them, what they mean. And honestly, I know it's just super easy to get overwhelmed with this unit. You'll be fine. And a lot of these questions, we put in a bunch of practice questions that Yago will go through with you. And all of this should help you with the problem set. So I think you guys are getting a good start on it by being here because a lot of people probably haven't looked at it yet, which is fine, long weekend, whatever. But I think this will help you get a start. So we're gonna go through every single pathway. So there's like, what's one? Like the RTK, there's the MAPK one, there's the STAT,
GPCR.
yeah, that one, all of that good stuff. We have all of them in here. So, like on the exam, that's a good question.
I think so. I think I think you get diagrams, but not everything is labeled.
Yeah,
I think.
yeah. I think that's the most you'll get. But yeah, I wouldn't be surprised if she just gave you it, but without the labels. Yeah. Okay, so here's what we're gonna do. We're gonna have the principles of cell signaling, so all the basic stuff that she talked a little bit about in lecture. We have each pathway, so I'll be doing the first two. Yago's gonna help you out with the second two, and we have like a bunch of practice questions in it. If you have any questions, like you don't have to wait till we're done, just like raise your hands, kind of chill. I was saying this earlier, but if you just came in, we are making a goal to like kind of be out by like 8:15, but if you have any more questions, I will stick around for a little bit. Okay, so principles of cell signaling. So this is we're gonna cover the basic stuff first, and we're gonna build our way up to the pathways. Okay, so first thing, cells receive signals from their environment for different reasons. If a cell wants to do a certain job, they might get a signal to do that job. Maybe it's a signal that's telling it not to do a certain thing. We'll talk about that. So like survival signals. If a cell is receiving a survival signal, what is it telling the cell to do? Survive, right? And we'll get into that. But one thing I really want to focus on, and she did a question like this in class, was the type of signals chemically, their chemical properties of what can get through and what can't. So we have the hydrophobic signals, which diffuse through the membrane and can be received inside the cell. Those are usually small and nonpolar. And then we have the hydrophilic. So somebody remind me, what does hydrophobic mean? Do we like water? We hate water. Hate water, right? So hydrophilic means we love water, right? So hydrophilic can't go through the membrane because the phospholipid bilayer, we'll talk about that in a second, and it interacts with the membrane receptor. That's like TLDR, but we're going to go a little bit more in depth in that. So let's quick refresh. So if a signal can pass through the phospholipid bilayer, it is blank and blank. Can somebody tell me? Yeah. Yep. And then so if a cell can't pass through, then it is right. So let's look at the diagram real quick. So this can kind of confuse people. So phospholipid bilayer, lipid is fat, right? So usually when I think of like hydrophilic likes water, means it doesn't like fat. That's kind of how I've always thought about it. Like hydrophilic likes water, doesn't like fat. Hydrophobic doesn't like water, likes fat. Does that make a little bit of sense? So we have the two hydrophilic tails, the ends, which are interacting with the aqueous solution. So we like water in that case. That's why those ends are like that. In the middle, though, it's all lipids. And so if you get confused, you're like, well, when we start talking about lipids, we're talking hydrophobic and hydrophilic. Just remember, if it likes water, it doesn't like fat. So if we have a smaller molecule that is hydrophobic, so which means it likes the fat, that means it can move through. So this diagram was really helpful to me when I took this class, just because she'll like sprinkle that question in here and there. I think there might be something like that on your problem set, which is like the properties of signals. If she says properties, like chemical properties of the signals and stuff, don't overthink it. Can it get through the bilayer or can it not? Do you have anything to ask that?
Just think, so the heads are hydrophilic, tails are hydrophobic. What part of the membrane is mostly what? The biggest portion of the membrane is hydrophobic. So that's the hardest challenge to get through. So you need hydrophobic things to get through that hydrophobic region. And obviously small, because the gaps in between are small. So you can't fit something large in between there. The smaller, the better.
Yeah. So there are two outcomes of cell cycling. One of them is you can alter protein function or you can alter protein synthesis. We're going to talk about what each of them means. And these aren't mutually exclusive, meaning these can happen. Like if one happens, it doesn't mean the other is not happening. So signals are complicated like that. You can get multiple different outcomes. There's multiple different things that they can do. It's never usually one or the other. So if she asks you anything about if these are mutually exclusive, answer is no. There's a lot of different possibilities. So let's talk about what each one means, right? So here's an example, acetylcholine, right? In a heart muscle cell, it causes decreased heart rate and force of contraction. The same signal, acetylcholine, in a salivary gland cell says that there's secretion of saliva. So these are obviously two different things that it's doing, but it's the same signal, right? So that's what I'm trying to say. So same signal can lead to different outcomes. So can somebody tell me, why might protein synthesis, and when I say protein synthesis, I mean changing from the root level, like changing the DNA and RNA of something to have different types of proteins. Why might be, protein synthesis. Oh, I think I wrote this wrong. Okay. Why might altering protein function, so the job of what a protein does, why might that be a faster way of making an outcome occur?
Synthesis. Oh,
Is it?
you said
No,
function.
it's function. Yeah, I wrote the wrong. Yeah. So protein function, yeah. Yeah, so the protein function. So I made a typo here. It's actually why might protein function. I'll change that later. If the proteins are already in the cell, all we have to do is like tell it to turn on or off or like change its job or what it's trying to do. Protein synthesis, so the making of proteins, we have to go through transcription, translation, all that good stuff, which takes time, right? So in this case, changing the existing protein function to make an outcome occur without having to like change the underlying DNA, genetic code, whatever makes these proteins do these things, is a lot faster than having to start from scratch and like make different proteins. Does that make sense? So like post-translational modification. Can somebody give me an example of like a post-translational modification? Yeah, phosphorylation, right? Adding a phosphate to something. That can be something like a way of turning something on and off, right? That's the switch mechanism we talked about a couple units ago. That's a pretty fast way to get a different response than going from scratch and trying to make the protein do a completely different thing, right? So that's the reason of why it's faster. So let's talk about blah, 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 cell death. Okay, so we're gonna talk about survival signals in a second. Are we all familiar with the term apoptosis? Do we all kind of know what that means? Apoptosis is cell death, programmed cell death. So if a cell is not receiving survival signals, it's going to go straight into the apoptosis or the programmed cell death. That's just what it's going to do by default if it's not told to survive. Okay, so that's what apoptosis means. I just say that because she doesn't specify what it means that much. She just refers to it. But if you don't know, cell death, don't get a survival signal, you're going to go straight into that programmed cell death. So we're going to talk about a couple of pathways. But first, I want to talk about signal amplification. So what I like to think of it as is signal amplification is gossip. So if I tell Yago something and Yago tells somebody else something and they tell a bunch of people, that message is being spread, right? That's like gossip. That's exactly what signal amplification is. So one thing tells another thing something, that tells a bunch of other things something, and now that signal is spread across like, a lot more. And that's like kind of in this diagram you can see. It's phosphorylation of multiple kinases. We'll talk about that. And then crosstalk. So when she says crosstalk is just one pathway or one, part of a pathway talking to another. So they communicate, they coordinate in this giant diagram that she shows you. I'm not a huge fan of it because it kind of confuses me, but all of the horizontal arrows signify crosstalk. That's what that means in this diagram. But so we can do a lot of different things with crosstalk. We can tell another pathway to stop doing something. We can tell it to keep doing something else. We can turn it on or off, but the whole purpose of it is to coordinate responses in the cell. So somebody tell me, then we talked about this a little bit in class. I know it's on your problem set. What are the pros and cons of having a multi-step pathway compared to a pathway with few steps? Let's start with the pros. Can somebody tell me some pros of having more steps in a pathway? Yeah, we can talk to a lot more people, right? We can spread the message. What else? More opportunities for crosstalk, right? Can anyone think of any others?
It's like there's like to be slower. It's like because you have to keep going like step and then step. If it's like multi-step out, like maybe if it's about two step is like quicker.
Yeah, so yeah, for the cons, right? If there's a lot more steps, that's gonna be a lot slower response than if we had a couple, right? Okay, let's move on to cons. What are some cons of having a lot of them? So we know that it's slower, right? So if we're trying to get a message across, it's gonna take a lot more time compared to a pathway with fewer steps. What are some other cons? Anyone think of any? Hmm? Yeah. That I is it like, are you talking about the one with more steps or the one with fewer steps? Fewer steps, right. So fewer steps is like, it's pretty fast. We can't really change a whole lot that's going on there because there's not that many steps. Anyone else have any more cons for having a multi-step pathway? Okay, I'll just show you. So we said pros, more amplification, right? Each step, we can talk to more people, we can spread the message farther, and there's more opportunities for crosstalk. So more ways that a pathway can talk to another pathway, intervene, do as it will. Cons. So slower signals, right? If you ever see the phrase diffusion rate limiting kinetics, it sounds fancy. It's actually not that fancy. All that means is the likelihood that two specific molecules are going to bump into each other when they're just floating around. That's all it means. So the slower signals, each step is slowed down by the diffusion rate limiting kinetics. So how likely is it that two things are going to bump into each other? That might slow us down between each step. So if she ever like mentions that term, that's what that means. So again, another question that we did in class, but I just want to go over now that we've kind of like gotten through the basics. If you don't know what endocytosis means, can somebody remind me? What is endocytosis? Bring things into a cell, right? So endocytosis of the receptor is the receptor's on the outside, we bring it in, it's now on the inside. That's what that means, in case you need a refresh. So let's go through one by one. So a signal has been transduced, and now the cell wants to decrease the amount of signal transduction. So we want to turn off the outcome. We don't want the outcome to happen, or we want to have less of that outcome. Can somebody tell me for just number one, would degrading the signal help us turn down that outcome? Yes or no, and why? How about we get like a thumbs up or thumbs down? Everybody thinks that would work? Thumbs up? Okay. Anybody want to say why?
You don't have the same signal then the like transduction and then the following like implications when replacing like something to start it and it's a signal that like I need this pattern.
Yeah, so less of the signal, less messages being passed, less of the outcome happening, right? So that's number one. Endocytosis of the receptor, number two. So again, we're bringing the receptor that's on the outside, bringing it in. Yes or no? Is this a good method? Yes, because if the signal can't bind to the receptor, How are we going to get it to keep going? Right? So this is a good way. Yes.
Signal and receptor is always like it can only really like cause transduction when the receptor is out there like accepting the signals, while like for the hydrophobic ones, like they don't need to have the same like the receptor outside, they just like diffuse into it, so it doesn't need receptor.
They need a receptor eventually.
Yeah, but is it like receptor inside the cell?
Yeah.
Yeah.
So what two is saying is that basically there's no receptor. The receptor's been taken away, basically. So without a receptor, you don't have the pathway anymore, right?
Is it still possible to get, signal amplification happening even if we don't have a receptor? Technically, yes, if we don't even have a signal because of crosstalk, right? Again, that's where crosstalk comes in. So if she ever asks you a question that's like, is it possible without a signal if we can do this and this? The answer is no, like, there's no receptor, nothing for the signal to bind to, nothing for the process to happen, but there is always, always, always the possibility of crosstalk. There's always a possibility of another pathway being like, actually, you know what? I do want this. I'm going to activate it for you within the pathway. So she likes to include that little like asterisk, like, no, but there's always a possibility for crosstalk. Okay, let's do number 3. So degrade one of the signal pathway components. Is that a good way of turning down a signal? Thumbs up or thumbs down? Thumbs down. Okay, so it's actually thumbs up. So this is also a good way. And the reason is during signal amplification, let's go back to that diagram. If we have less of the people that we're talking to, then we have less people that they're talking to, right? Because there's less people spreading the gossip, essentially, for those components. So degrading one of those in the pathway is a good way of turning down that outcome because the message is being spread a lot less. Does that make sense? Nods, shakes. Okay. Okay. I just, you know what? I'll show. So all of these. So let's go over number 4. So another pathway shuts off gene expression of the original pathway. What is this called? What's the term? Crosstalk, right? Another pathway doing something to a certain pathway, that's crosstalk, right? So there's always that possibility. That is possible. Another pathway directly shuts off one of these signaling components. Same thing. So it's basically just a repeat of what we're saying earlier, which is the message can't spread. And then in these numbers, we're talking about the cause of why it's not spreading. So 5, one pathway is like, you know what? I'm going to shut this off on a different pathway, one of the components. That message is probably not going to spread, right? So crosstalk always, always, always is a possibility. It's never fully no. It's no, but there is always a possibility for crosstalk. Does that make sense, guys? She's gonna hit you with the crosstalk asterisk. So, okay, pathway time. Everybody, deep breath. It's not as complicated as you think. Okay, so let's talk about step 1. So, step 1 is signal binds to the receptor domain, which is the portion outside the cell that binds the signal. So, signal's right here. Can you guys see yeah, you can see my cursor. And it binds to the RTKs. Notice how the RTKs are separate at first. They're separate at first. They're not together yet. They're just chilling. Signal binds, and it allows the two of them right here to come together. And that is called what? Dimerization, when two things become one thing. Okay, and it's not just limited to RTKs. This can happen in other things too. So signal comes in, binds one of the RTKs, they come together, and when they come together, what happens? Yes, but what causes the cross-phosphorylation? Why can we now do that? Because there's a conformational shift happening. Can somebody remind me what conformational shift means? Yeah, change the shape. So something binds something else. The chemistry of that thing is changed because the structure of it has changed. And what do we know about structure? It determines what? Function, right? So you change the chemicals of something, the shape changes, therefore the function of it now changes. So signal comes in, 2 RTKs bind together. Now it allows it to cross-phosphorylate each other, which means they add a phosphate to each other, right? And this doesn't always mean that we're turning something on. I really want you to get that in your head. Adding a phosphate to something does not always mean that we're turning something on. It's just changing the chemistry somehow. Oh, okay. Okay, it's back. It could also be turning something off, right? all it's doing is changing the chemistry. Usually when we talk about it, it's turning something on just because the pathways that we're talking about, that's what happens. But just know, it's not an end all be all. It doesn't always mean you're turning something on. We're just making the shape change, which means we're taking the function change. So come together, phosphorylate each other. Please. They cross-phosphorylate, and then the phosphorylated tyrosine kinases serve as binding sites for the downstream proteins. And this is what we call the signal transduction cascade pathway thing. It's like just a domino effect, basically. So this is the fun stuff. This is the fun stuff. So yes. Does anyone have any questions on this actually before? Yeah.
Of the receptors, so that they phosphorylate the tyrosine.
Yeah, so wherever you see a phosphate, wherever you see the yellow P's, that's where there's a tyrosine in the protein structure.
Yeah.
And that tyrosine is what can be phosphorylated.
Yeah. And also, does anyone know what RTK stands for? It's receptor tyrosine kinase, right? So that's good to know because Yaga will talk about that later, like the difference between the domains and stuff, and like we'll talk about there's questions that are like, what happens if it changes and like to something else that's not tyrosine? We'll talk about that. But RTK, receptor, tyrosine,
And
kinase.
kinase is important because kinases phosphorylate, right? And they auto-phosphorylate each other. They cross-phosphorylate each other. If they weren't kinases, they wouldn't be able to phosphorylate each other.
So the two are, the signal comes, then the two RTKs harmonize, and that makes a conformational change, and that causes cross phosphorylation of each other.
Exactly.
Yes. So whenever you're like something activates something else, and in the topic note, she does it in a really long way because she always emphasizes, she's like, this binds to this, which then causes a conformational change, which then causes things to be activated and things to keep going. So always specify the conformational change because as we know, structure determines function. Also, if you see autophosphorylation, that's just another way of saying cross-phosphorylation. It just means it phosphorylates itself. Okay.
And then those phosphates end up being binding sites for those proteins that you see binding to all the P's, which will be important later on.
Okay. GRB2. GRB2 is this dark blue guy right here. That's the adapter protein. Okay. So it comes in, it binds to one of the phosphorylated tyrosine kinases on the RTK through the SH2 domain. Yago is going to talk about the difference between the SH2 and SH3 domains in a bit, but just know that for now. It binds to the SH2 domain on the RTK. Make sense?
Yeah.
Yes. So one of those phosphates that we just added on, phosphorylated tyrosine kinases, that this fun part. We're adding GRB2. So GRB2 binds, and when she says RAS-GEF, in this case, we're talking about SOS. This is the SOS protein. So this is that RAS activating protein here. This is SOS. Okay. So GRB2 binds the RAS-GEF right here through the SH3 domain with PXXP motif. We'll talk about that in a second, which activates this. So GRB2, the dark blue, has an SH3 domain. The SOS has the PXXP motif, like hypothetically, like right here. Yago, do you want to like explain a little bit, but like how they bind each other? You were telling me this earlier.
Yeah, so basically right here is, you guys know what a domain is, right? It's a part of the folded protein. So right here will be an SH3 domain on the adapter protein, right? And then there'll be a PXXP right here on the RAS activating protein. And those basically bind together like puzzle pieces. I'll go over it more later in a later slide. But those domains are just like what goes together with what. And then again, here would be an SH2 domain that allows that to bind there.
So that SOS protein, aka it's labeled RAS activating protein here. This is the RAS-GEF, is going to change RAS from GDP to GTP through conformational changes. So this is the asterisk. When RAS is in the GDP bound state, it's off. And when it's in the GTP bound state, it's on. So similar to like ATP and ADP, right? Like how you kind of switch between those to turn things on and off. This is the same concept.
And the GEF, the RAS GEF is the same thing as the nuclear import and export. You guys did same thing. You have the gap that changes from GTP to GDP, and you have the GEF that does GDP to GTP. So it's doing the same thing here. Its sole purpose is just changing the RAS protein that has GDP to GTP. Make sense? And when that happens, it's not like actually switching it. The GDP just gets kicked out. So it goes that way. And then free GTP comes in and takes its place.
So quick recap. This is which protein? What's the dark blue? GRAB2, right? This is GRAB2. This is the SOS or the RAS-GEF SOS protein. And just like Yago said, we're not getting rid of this guy. It's just being let go. GDP can now diffuse in, activating that RAS. So this is the end of this kind of part of the pathway, is the activated RAS protein. So now let's talk about yeah.
I'll explain it.
Yeah, he's got all his slides about it. So he'll cover them.
It's similar to SH2, SH3. I'll talk about that too.
So, okay, now we have the activated RAS, which has GDP or GTP now. GTP. Remember, when RAS has GTP, it's on. It's active. Okay. It causes activation of a thing called MAPK3. We don't know how. It just is. Okay, so don't have to know that. And it causes this thing called the MAP kinase cascade. So just know this. Active RAS activates MAPK3. And remember, is RAS active when there's GDP or GTP? GTP. Okay, so it's really important not to get these. It's so easy to get these confused in your head. Just know, if I know that GTP has to be there when RAS is activated, you don't have to try and remember like everything. So MAPK3 is now active. Again, we don't know how RAS does it. It's just active now. It can now bind ATP and its substrate, MAPK2, just same as last time, right? Things activating each other. MAPK2 now shifts conformation. Now it's active. Now it can bind ATP and its substrate, which is MAPK. And finally, the phosphorylation of MAPK activates it to phosphorylate downstream targets. Remember me, what do kinases do? Phosphorylate. It's just a matter of domino effect, things phosphorylating other things, and then we have an outcome. Okay, and the outcome of this is MAPK is now active to phosphorylate its downstream targets. And it can be transcription factors, but it can also be other stuff too, to make something happen. Anybody have any questions on this pathway?
For the second part of this pathway, an important thing, I think when I took the class, it was on the exam. So remember, the first step right here, this is just activating, not through phosphorylation. So you see how there's no P's on this MAPK3? That's not phosphorylation. Again, we don't know how it's activated, it's just activated. But then each further one is phosphorylated to activate it. And so like I talked about, a kinase phosphorylates, right? So the first one's called kinase.
No, it'll come back.
Because it is the kinase for kinase. Does that make sense?
So why is there two phosphates on kinase and then just kinase? If they're like, shouldn't there be three on kinase?
It doesn't give all the phosphates. It just basically needs two phosphates to activate it. But you don't need to like know that much into detail. Did it make sense though what I was saying about? So like for example, if you have MAP kinase, just one kinase right here, right? To activate it, you would need the MAP kinase. Does that make sense? It's the kinase for MAP kinase. And then the kinase for that would be the kinase.
Yeah.
Just remember it goes opposite order, 3, 2, 1. All right.
Yeah.
I think she quizzed us on that when I took the class as well too. So first step, just activated, not phosphorylated. Then further down, each one is phosphorylated, and the number of kinase goes down until you get to the end where it's the actual proteins, and it could activate a bunch of different types of proteins, like just changing the activity or the actual protein synthesis. Make sense?
Yeah. MAPK3 is just a fun way of saying map kinase. So yeah, that was good. Yeah, remember 3, 2, 1. Okay, question. So, in which of the following would you expect to see unrestricted growth and increased cancerous phenotypes? Don't raise your hand right now. I want you guys to talk about it for like 30 seconds, a minute maybe, and then we'll come back to it.
Remember what happens in a cancerous phenotype: uncontrolled signaling, like nonstop, always on.
Anyone have any guesses? You said 3, 2, and 3. What else? What else? Anyone else have a guess and why maybe?
Stepping like or not like.
Oh, it's like maybe like
I'm not
this.
touching.
It, so when you like.
I'm not touching it though.
Okay, then you're not-
Since it does it.
It's all your fault. Okay, guesses. Somebody raise their hand. Give me a guess. Give me a guess. Anything, guys. Yeah, like what's your answer? Like so it's one of them. Yeah, but we'll go through each one to like explain why. 3, group consensus. Okay, well, we'll go with 3. Yeah, so that's the right answer. So let's go through each of these, right? So what Yago said, cancerous phenotypes means unrestricted cell differentiation, cell division, right? That's what cancer is. So the signals keep differentiating and keep dividing cells. So everything is always on. So GRAB2. GRAB2 has a mutation such that it's unable to bind to the phosphorylated tyrosine kinase. So if GRAB2 can't bind to right over there, is the signal going to keep going? No. I mean, again, always possibility for crosstalk, right? But it wouldn't be able to keep going. So that's number 1. Number 2, RAS has a mutation that causes it to be constitutively bound to GDP. So that means it's always bound to GDP.
That would be cancerous, no?
That's what I thought. That's what they have in here.
Yeah, so that one would be correct as well.
Okay, I lied, guys.
Because if it's always bound to GTP, it's always on, right? So the signal's always going forward, and all this is happening. I don't know why.
Yeah, I don't know when they have. Oh, let's go back. Okay, and MAPK3 has a mutation that is always activated. So MAPK3 activates what? MAPK2, which activates what? Just downstream proteins, right? Like now it can do its job. So if it's always activated, which means it's always going to activate MAPK2, which is always going to activate MAPK3, we're going to see a lot of cell differentiation, cell division, right? So when she says cancerous phenotypes, that means unrestricted signaling. It means things are overactive. There's too much cell differentiation, too much cell division going on. Make sense? Sorry about that. I did not catch that. Okay, survival signaling. So what is apoptosis? So what do we need to stop apoptosis? What kind of signal? Survival signals, right? So signal, sometimes I don't know if to say dimer or dimer. I'm gonna say dimer. And what is a dimer? It's just like two things that came together as one, right? It binds to RTK. We know RTK. The receptors dimerize, cross-phosphorylate, right? We already went over that. So this guy, the blue guy, PI3 kinase binds to the phosphorylated RTK through the SH2 domain. Again, we're going to talk about that soon. Yago's got some slides on the different domains. It binds here, right? PI3 kinase now phosphorylates PI. This is PI right here. PI is the lipid that gets phosphorylated. Once we phosphorylate it, it's called PIP, if you're wondering what the difference is between those two terms. So PI is just before P, right? The phosphate. Yep, so as third phosphate creates PIP3. We just call it PIP, but she has kind of both in there sometimes. Again, so the blue, right, serves as the binding site for PK1. This is PK1. This is part of the same diagram, by the way. So AKT, this is the green, is the central regulator. That's what we call it. It's regulated by this guy and the purple guy right here. AKT binds to the PIP. PIP is the 3 block right here. So that there's 3 phosphates, blue block. That's what I'm talking about. And then PK1, this guy also binds it and phosphorylates AKT. Now, when this is phosphorylated, is it fully active? It's not actually. So usually we're used to seeing like, okay, something's phosphorylated, it's fully active, it's like fully doing this job. That is not the case with AKT. Come back. So when we phosphorylate AKT, it changes its conformation. We know that, which allows it to release from this guy and diffuse freely in the cytosol. But it's not fully active yet. So remember, when AKT is phosphorylated, when the green guy is phosphorylated, all it lets it do is release from this blue block guy. Okay? So once it's out of there, now PK2, protein kinase 2, the purple guy, can come in, phosphorylate it, fully activate it. So again, if she asks you, when you phosphorylate AKT, is it fully active? Yes or no? No. All it lets it do is get away from PIP, which then lets PK2 come in to fully activate it. Now, the ending of this pathway is not fully, but when AKT is fully activated, now we can talk about bad and all the other good stuff. So that's really important. So we phosphorylate it once, it gets released, we phosphorylate it again, now it's fully active. Does that make sense, that concept? Okay. So again, we need AKT to be active to keep the signal going. Now let's talk about bad in BCL2. So in this example, AKT phosphorylates bad. Bad is this guy. I just like saying that. It's the blue guy, right? Which lets it let go of this. So let's talk about that a little bit. So the red guy, this is the cell death inhibitory protein. So it's only active when it's not bound to bad. So this is great because when AKT phosphorylates bad, all it lets that do is let go of this red guy, the BCL2. Normally, they're always together. Okay? So when they come apart, when bad is phosphorylated, it inhibits programmed cell death, only when it's apart. So they're together, BCL2 can't do anything about apoptosis. Bad gets phosphorylated by AKT, they're apart now. Now we can prevent cell death from happening. It's easy to get mixed up in this, but I really want you to get this part. So just to go over it again. AKT, it's now activated because we phosphorylated not once, but twice. Comes in, phosphorylates bad, green guy. Green guy, let's go of, sorry, not green guy, blue guy, let's go of red guy. And now red guy can do its job of stopping cell death from happening, which means we survive, But remember, the default is always the cell death. If we don't do anything about this, the cell's going to go through apoptosis because it's just, they're stuck together. nothing can be done about the situation. It's programmed cell death. It's kind of like the default option. Yes.
This is the part about the first part about the AKTs and then like the green and brown.
Yeah. Yes. Let's go back. So, okay. So we end this slide with this guy right here. Okay. So PI3 kinase has now phosphorylated this guy. And now we have PIP3 is what we call it. We just call it PIP. Okay. Now, that same blue block serves as the binding site for PK1. But let's not even get there yet. The first thing that happens is AKT binds that blue block. Then PK1 also binds and phosphorylates this guy. So this guy comes in first, then this guy also comes in to bind. So green guy, AKT, comes in first, binds it, and then brown guy, PK1, comes in and also binds it, and at the same time, phosphorylates AKT. Yes.
They're two separate ones. It's the same type of protein, but two separate proteins. So if you have PIP, it can either bind AKT or PK1.
So recap: green guy binds, then brown guy binds to another one, and at the same time, it activates the AKT. Does that make a little bit more sense?
PK1 and then the brown guy and then which other one binds for the AKT.
Here, I'll explain the whole thing quick to you guys. I think I can make it make sense.
Okay. So you have a survival signal comes in, right? These were separated at first, right? Like before. Survival signal comes in because the cell wants to survive. They come together, auto cross phosphorylate, right? And now they're active. And they have the phosphate, so the PL3 can bind to that phosphate. Make sense? Now the PL3 is active, and that phosphorylates PI, turning it to PIP. Okay. And now you have active PIP. Okay, and that's going to serve as a binding site for PK1 or AKTs. Okay, so you have AKTs binding to some of it, and you have PK1s binding to the other ones. Okay, once you have both of those, PK1 phosphorylates AKTs, and that causes the AKT to bump off the PIP. Okay, and then PK2 comes in and does just what PK1 did. It phosphorylates it again, and now it's completely active. All right. So it's released, free in the cytosol, and active. Does that make sense so far to there? And then Anjali will go on to talk about it more. But once that's active, it phosphorylates bad, which was holding on to BCL2. Once it's phosphorylated, the phosphate takes the place of BCL2. So now it's holding a phosphate. It can't hold BCL2 as well. So it releases it, and BCL2 is cell death inhibitory protein. So once it's free, it inhibits cell death. So the cell stays alive, doesn't die. Does that make sense?
Yeah, that was a good recap. Anyone have any more questions about this pathway before we move on? Okay. So yeah, this is just the full picture. Again, all these are going to be sent out, but the first couple slides, how they're like cut up, it's all part of this. So let's do, I think this was in class today. Again, I'll let you kind of like do this on your own. PI3 kinase is a blank that is phosphorylating a blank. Think about it for 30 seconds, come back to me.
PL3 kinase remembers this one right here.
Okay, does anyone have the answer for this one?
But is a protein that's phosphorylating to lipid.
Yeah, perfect. And
remember that you have the protein right here, the kinase, and this is the lipid, the PI. and then it gets phosphorylated in this step right here.
And just for a little bonus question, I mean, Yago will talk about domains in a second, but PI kinase has which type of domain? Does anyone know? Yeah, exactly. For some reason, this is in the topic notes and she talks about it in class. I don't know if she'll have a question on it or it's in your problem set, but that is an important distinction to know. Is that it's a protein that is phosphorylating a lipid, which Yago said was PI turning into PIP. Are we feeling okay? A little bit better about this pathway? A little bit? So, the question that somebody asked before about, okay, what if it's not a tyrosine? What if we change to something else? Does it always have to be tyrosine? This is kind of a question related to that. So let's say you have a question that's like, how are we going to figure out which SH2 domain is binding to which phosphorylated residue amino acid on the RTK? So what does RTK stand for? Receptor tyrosine kinase, right? So let's say we change that to an alanine, which is hydrophobic, and everything else functions. So what is not binding to RTK anymore? That's how you find like what the binding partner was. This is kind of like a bonus slide. It's basically saying that if we want to stop what's happening, a way that we could do that is by changing the tyrosine to something else. So breaking that pathway, not letting the signal go any further.
Yeah, so if you remember in the first pathway we talked about, there was 3 phosphates, and these there's only 2, right? Because the one that was up here, it was a tyrosine that could be phosphorylated, got changed to an alanine, now can't be phosphorylated, so there's no phosphate there, so the protein that was binding to that phosphate can't bind anymore. So if you want to see, I think it was grab, right? Grab was this one? Yeah. If say we wanted to see which one of the 3 grab bound to, we could take away 2, change them to an alanine, they can't get phosphorylated, and we see, is GRB still binding? If it's still binding, we know that that's the SH2 domain that it's binding to. If we took that one away and kept the first one and saw that it wasn't binding anymore, we would know that it wasn't binding to the first domain. Make sense? So that's kind of how they figured out which domain each protein was binding to. Also. All right, so a little bit about the different domains. So an SH2 domain, these are the proteins that bind to the SH2 domains. We have the GRAB2, STAT, and the PL3 kinase. And what these domains basically are is tyrosine that could be phosphorylated. Okay, so if you see tyrosine, SH2. Okay, SH3 is not phosphorylatable tyrosines. And a protein that has an SH2 binding domain has a domain that can bind to SH2. Okay, so if, like with GRB2, it has an SH2 binding domain. It has an area of it that can bind to SH2, and SH2 would be a tyrosine residue that's phosphorylated on another protein. Does that make sense? The SH2, it has the SH2 region, but the binding domain, the SH2 binding domain, is on the protein that's binding to the SH2 region. Make sense? And then SH3, again, GRAB2 has that one, but on the opposite side. Remember we talked about that? I don't know if there's. Here, I'll pull up a picture quick. So it has 2 sides, right? Here is the SH2 binding part, and the SH3 binding part will be on this side. Okay, so don't get confused just because it's the same one. It has both because one on each side. All right. Yeah, I'll show you guys that, but the next one I'll talk about is over here on SOS on this side. Okay, so SH3 is different. It's not tyrosine. It's on the other side of GRAB2, and it's what binds to the SOS. And that part of the SOS is the PXXP. motif. So think about SH3 always binds to the PXXP. Makes sense? Those two always go together. Yeah, that's basically it. This is the most like detail you'll have to know about those. Just know which goes with what, which have which, and what it means, kind of. Good. Okay. Question. What would cause the RTK PL3 kinase AKT pathway to remain on or off? Here, I'll bring it. This one. What are some things that could make it either stay always on or always off? Right here. This. Yeah. So a gain function in this would do what? Exactly. Yeah. So if you have this always on, this always gets turned on, and everything further stays on. What else? How about something that could make the pathway always be off? Yeah, right here. Yeah. So loss in function of this wouldn't be able to phosphorylate this, which wouldn't activate it or release it or anything further down, right? All right. Those are good examples. Then there's also, so basically, gain functions early on make the signal stay on. Loss of functions make it stay off. Except when you go further down, because say you have a gain function in bad, that would mean it's always holding on to BCL2, which would never be released, which would never stop cell death. So these ones are the ones that are kind of tricky, like gain of function, loss of function. Here, gain of function always on, gain of function on, gain of function on, gain of function on, loss do the opposite. Here, it's kind of swapped. Does that make sense?
So like a gain of function of bad would just maintain the photosis. But
Yeah.
if you had a loss of function in bad, it would basically just be like doing the same thing.
Yeah, then it wouldn't be able to hold on to this, and this would be released. And then same thing, you have loss of function, BCL2. It can't stop cell death. So then, yeah. Okay. What would happen if AKT had a loss of function? Yeah. So if this can't work, it can't phosphorylate bad, which can't release BCL2, which would mean cell death or apoptosis would always happen. This gets tricky because whether or not it's stopping or like it, for me, it got confusing being cell death inhibitory protein that inhibits cell death instead of like causes cell death. So just make sure you get clear on that. Okay. JAK STAT pathway. Again, we have the same two separate receptors. A signal come in this case, it's prolactin. All right. They come together, and now there's a part of the receptor called the JAKs, got JAK1 and JAK2. Those cross phosphorylate each other. All right. So we see JAK2 phosphorylates JAK1, JAK1 phosphorylates JAK2. All right. That's when the receptors get brought together. Now, those each phosphorylate their part of the receptor. Okay. So these were activated. Now they're phosphorylating and activating the receptor. Those phosphate groups on the receptors, again, act as binding sites, like we saw in the other channels. And STAT5s bind to that. Okay. And once STAT5 binds, Then again, the JAKs phosphorylate the STAT5s. So first it just binds to that phosphate, doesn't have this phosphate. Now the JAK comes and phosphorylates it, so it's got the two phosphates on both sides, all right? Once they have those two phosphates, they form a dimer, they come together, and that dimer gets released and goes on and regulates transcription. In this case, prolactin, transcription of milk protein genes. Does that make sense? Any questions on that? STAT, again, STAT is binding through SH2 binding domains because it's a phosphorylated tyrosine, right? Okay, question. What would be the effects on the JAK-STAT pathway if there was a loss of function mutation in the phosphorylatable region of the JAKs so that those regions can't be phosphorylated? So you have a loss of function in these JAKs so that they can't be phosphorylated. What would happen? What was that? Exactly. So the whole pathway would be stopped, right? Because if these can't be phosphorylated, they can't cross phosphorylate each other, then they can't phosphorylate the receptor. STAT can't bind, STAT can't be phosphorylated, and STAT can't form the dimer. So it stops it right here. In between switching these two steps, you can't get the phosphate. Does that make sense? Okay. Exactly. All right, another question. What if there was a gain of function mutation in the phosphorylatable region of STAT5? So here's STAT5. This is the phosphorylatable region where the JAKs are phosphorylating it. What if there's a gain of function in that? What would a gain of function mean in that situation? Exactly. So if this is always phosphorylated, it doesn't need to be activated by anything before because gain of function, remember, means always on. They would always be phosphorylated, always form that dimer and go on further in the pathway without needing any of this before. The stats don't need to, the JAKs don't need to phosphorylate it. Make sense? Okay. Next pathway, GCPR, G protein signaling. So GPCR is just a collection of proteins basically all together. You see right here. We have the transmembrane part. So if this is the membrane right here, outside of the cell, inside of the cell, you have a bunch of regions that go in and out of the membrane. And then you have G alpha, G beta, and G gamma. Alpha is A, beta is B, gamma is Y. Okay? Those are all kind of together. Then you can have something come in and bind to the part of the receptor. And what that causes is that causes the separation right here of G alpha from beta and gamma. So you see right here, G alpha is alone, and then beta and gamma are alone. Beta and gamma always stay together. Alpha is always alone when it's separate. All right. And then you go on to here. This had GTP. So when the ligand came in and bound, you have GTP bound to G alpha, and it hydrolyzes that GTP to GDP. So we see right here, the GTP is getting thrown out. In this, remember when it was GTP bound state in the other pathways we talked about it, GTP means on. So it was on when it was bound to GTP. Right here, the ligand got bound, they separated, it's bound to GDP, it's active. Okay? So this is the active step. We see there's signaling coming from both the beta and gamma and the alpha proteins. When it does the GTPase activity and hydrolyzes GTP to GDP. GDP means inactive. So now it's inactive. When it's inactive, it comes back together with beta and gamma and forms that one complex again that we had at the start. Okay? So remember, it's important to remember alpha is the one that does the hydrolysis, which makes it a GTPase, right? A GTPase is a protein that changes GTP to GDP. And then these two just separate. They still contribute to signaling, but we don't talk about the exact signaling in this class. And then another important thing to know, do you see this little line on G alpha and the little line on gamma? Those are just lipid anchors. It's a part of the proteins that allow it to stick to the membrane. And so it can move alongside the membrane, but it won't be going into the cytoplasm wherever it wants. Okay? This is just more about it. Everyone good with this pathway so far? Where do you see R? Oh, right here.
Regulator of G proteins.
It's just like a helper protein. I don't know exactly what it does.
Yeah, I don't think she'll talk too much about RGS specifically, but
Yeah.
Or things like do things. They're bound to alpha and
What?
of the effectors.
yeah, you just do things later to.
They just help the reactions. The like say hydrolysis, say here changing GTP to GDP takes time. The effector would just like help it do that or like help it pass its signal on.
Yeah, well, because they're enzymes, right? And what enzymes do is they like lower the amount of energy you need for a reaction to happen. So without them, you could still do it. It'd just be like really slow.
So the signaling and clock, like. Once they're separated, and then once
They're active. Yeah, there could be infinite number of downstream components. A lot of like a GPCR is basically a type of signal, a receptor, and any pathway could use that type of receptor. No, it's just gonna send a signal down to another protein, and then it's just gonna go on and on. But you don't need to know. You're just studying the GPCR part, not the downstream effects on this one.
So like usually like binds is something impossible.
Yeah, I know what you're saying. No, but yeah, no, this one's different. So it's just the signal comes in, binds, separates the two. This is bound to ATP, so it's active. They're both separate, so this one's active too. They just send their message, whatever the message is, down. We don't know exactly what it is in this situation. And then gets rid of GTP, changes it to GDP, inactive, and they come together. That's all you need to know for this one.
Yeah, just looking at like different ways that pathways like can send messages basically. Like before we saw like phosphorylate, and then this one it's like a little bit different of like a mechanism, right? Because like they're coming together, they're apart, it's like a machine a little bit. So we're just looking at the different ways that.
Okay, what are the three subunits of the GPCR, and why are they subunits and not domains?
Let's start with the first one, too. Oh, or
Is it not alpha, beta, gamma?
Alpha,
And
beta.
they're, subunits because they're individual, like, polypeptides.
Exactly. So it's alpha, beta, gamma, and subunits are single polypeptide chains, a separate one. A domain would be all part of the same protein, but independently folded parts. And they're all separate. Make sense? Which subunits have lipid anchors, and what's the purpose of those lipid anchors? alpha and gamma. And then what's the purpose? To anchor them, but why would you want them anchored? Basically, yeah. So right here, remember I talked about the anchors. Those are parts that can interact with the membrane, anchors them to the membrane. We want them to separate to be active, right? So we want them to be able to move sideways. But if alpha could just move into the cytosol, then it wouldn't be close. It wouldn't be able to go back. It could go far away from the next protein that it wants to signal. So the whole pathway would just get messed up. We kind of want them to be able to move around a little bit, but not too much. Perfect. Which subunits have the ATPase activity?
Want to specify like what that?
Remember, ATPase means you can change ATP to ADP. No.
He's gonna answer.
Just one. Alpha. So the beta gamma together still contribute signals, but they don't have the ATPase activity because they're not changing. I mean GTPase. That's what I mean.
Yeah.
ATPase would just change the ATP to ADP. GTPase would change GTP to GDP. This one would be wrong. It'd be GTPase. Does that make sense? So here, this is the one that's doing the GTP hydrolysis. Here, it's still contributing to sending a signal, but it's not activated and inactivated by GTP and GDP. All right. Out of all the pathways we talked about, which do we think has the most opportunities for signal amplification and crosstalk? So I put pictures of them here. It might be kind of hard to see. So I'll do 1, 2, 3, and 4.
And also, what the pathways that have the most signal amplification and crosstalk, do they have more or less steps?
So which has the most steps? This first one, right? Because we have this step, this step, this step, and then all these, right? Each one of these steps could activate a bunch of different ones. Each one of these steps is an opportunity for a different pathway to come and input something. Like if you have the GPCR one, there's basically only these 4 steps. Less chances. Yeah. Yeah, but in this situation, we're just talking about the beginning. Yeah, but in this section of it. Yeah. So remember, more steps means more opportunity for amplification and crosstalk. That's all. We can stay for any questions on this problem set or anything else in the class.
Yeah, thanks for coming, guys. We sent out the recording and the slides. Yeah, actually, yeah, everyone knows it probably.
How do I stop the
I'll
recording?
stop it.
So, this is the most challenging topic I've come across.